My name is Steve Strauss. I'm a professor in the Department of Forest Ecosystems and Society and director of the OSU Outreach and Biotechnology program. Tonight's lecture is the second of 5 this academic year, part of the 4th year of the Food for Thought lecture series. The series focuses on biotechnology and related environmental, safety, and agricultural issues, and is sponsored by the American Society for Plant Biology, the Weight and Lois Rising Lectureship Fund from the OSU College of Agricultural Sciences, and the OSU College of Forestry. Dr. Felicia Wu is a faculty member in the Graduate School of Public Health at the University of Pittsburgh. She obtained her bachelor's degree from Harvard in 1998, majoring in applied mathematics and medical sciences. She earned her PhD at Carnegie Mellon University in 2002, where she majored in engineering and public policy. Dr. Wu is not a reductionist like me. Instead, in keeping with the theme of the lecture series, she brings a broad policy and risk perspective to studies of agriculture and food safety. For example, she is currently working on the relationship of molds to indoor air pollution and asthma, including on policy solutions to improve public health. She is co-director of a $2 million grant from the Gates Foundation to study the economic aspects of toxin reduction in maize and groundnut in Africa, with the goal of improving market access by the poor. Though she will address biotechnology in her lecture today, you may be happy to know that she is no gene jockey, and so is likely to avoid the genetic minutia that so often thrills audiences far and wide. Despite her youth, compared to me and some other folks around here, Dr. Wu has already obtained a number of forms of recognition for her scholarship. She started early as a National Merit Scholar and as a Jeopardy! Teen Tournament semifinalist back in 1994. Way to go. She was an Environmental Protection Agency STAR Star Fellowship Awardee in 1999 through 2002, obtained a National Institutes of Health Early Career Award as a multidisciplinary clinical scholar in 2007, obtained the 2008 Chauncey Star Award as the Outstanding Young Risk Analyst in the USA, and was recognized as a Sigma Xi National Science Honor Society Distinguished Lecturer for the current year. So we are indeed lucky to have her here. Today, Dr. Wu will speak on the environmental and food safety record of one of the most widely grown genetically engineered crops in the USA and the world, insect-resistant corn. She will give a broad biological context for this new form of maize and how we judge its safety, and also discuss the surprising yet growing evidence that, contrary to public fears about biotechnology in some quarters, this form of maize might be not only as safe, but it might be substantially safer than conventional or organic maize, especially in the developing world. The title of tonight's lecture is Improving Food and Environmental Safety, the Surprising Role of Genetically Modified Corn. Please help me in welcoming Dr. Wu to OSU. Woo!
What I'll be talking about today is, first, I'll give you some background of some of the pressing food and environmental concerns today. We're actually in a very interesting era in agriculture now that we're not only using food crops for human food and for animal feed, now we're even using it to power our automobiles and even our planes. So I'll describe some of those issues, and then I'll backtrack to a history of how we humans have domesticated corn for our own uses over the last 11,000 years. I'll go on to describe briefly how it is that we create genetically modified corn, and then the primary portion of this talk will be describing to you one type of GM corn, BT corn, what it is, how it's regulated in terms of food safety and environmental concerns in the United States, what the risks might be, and what the benefits might be in terms of toxin reduction. And then I'll wind up with a discussion of BT corn in the context of current events. So this is a slide that I normally present to my class. I teach 2 classes in environmental risk assessment at University of Pittsburgh. I say, well, what are the major environmental concerns that we've always been worried about? We are concerned about air, water, and food. Impacts to the ecosystem, impacts to human health, impacts as far as global climate change. They all revolve around what happens to the quality, supply, and production of these 3 very important resources. So we care about air quality. We're not necessarily concerned about air supply and air production. Those just aren't concerns. As far as water is concerned, we are concerned about water quality, particularly drinking water quality. Now, ever more, we are concerned about water supply, whether the world has enough fresh water supply for all of the human demand for it. And then as far as food is concerned, of course we're concerned about food quality, the safety of our food. Now we are concerned as well about food supply. This has always been an issue as far as our growing population. And we're also concerned about the environmental impacts of food production, that is agriculture. Now in the context of agriculture today, we care even more about food supply, as I had talked about. Now we're facing an interesting era in agriculture in which our corn and our soybeans are not just going to feeding us and to feeding animals, they're going to making ethanol and biodiesel to power our motor vehicles. That's one issue. Another issue is the fact that because there are so many competing demands for our food crops today, we're intensifying agriculture. And what is the impact to the environment? What's the impact to air, water, and food? And more than any other crop, corn is at the center of all of this potential controversy. So how did we domesticate corn, which is essentially the crop that we produce the most of in the United States? If you look at this photograph to the right, looking at that grayish stalk on the left, this is a grass. This is a species of grass called teosinte. Teosinte is a grass that's native to Mexico and to Central America. If you look at that, does that look appetizing to you at all? And yet it took some human genius 11,000 years ago. Well, first of all, you might notice that that blade of grass is actually separated into individual kernels. That 11,000 or more years ago, somebody actually consumed some of the kernels of teosinte and said, this is pretty good. We could potentially plant it in subsequent years as a potential source of our food supply. Now you might be wondering, so teosinte is actually the ancestor of modern day corn. You can see that, of course, modern day corn looks very different. It's golden, it looks luscious, it looks appetizing. So how did we get from the grass teosinte to modern day corn? For approximately 10,000 or more years, the main way in which we domesticated corn was to hand select the kernels of the blades of teosinte and all its descendants that seemed to have the traits that we desired the most. The ones that had the sweetest kernels, the one that appeared to have the best nutritional benefit, the ones that came from stalks of grass that appeared to be the healthiest. Maybe they had the strongest stems, maybe they were more drought resistant, maybe they had other desirable properties. And so in essence, what we do in agriculture by hand selecting in this way is that we speed up evolution of the species to basically meet our own human desires and demands. Now, hand selecting for desirable traits was the main way in which we accelerated evolution of corn up until the 1920s. Then in the 1920s came the whole advent of the idea of hybrid corn, and indeed other hybrid crops. Now, what exactly does that mean? No longer do we have to rely on corn to either self-pollinate or to pollinate with other corn plants immediately surrounding it, because what happens essentially is that the corn puts up its anthers and the pollen spreads, and at the same time corn also has silks that collect the pollen, and that is how corn reproduces. Now, for example, you could take pollen from corn growing in one end of your cornfield and mate it with corn clear at the other end of the cornfield. Or you could make corn from Iowa with corn from Missouri, or corn from Iowa with corn from Mexico or Taiwan. All you had to do was to control the pollination. And now you could even further select for traits that were desirable. For example, suppose that in the Midwest region, corn was produced with very sweet kernels, but maybe the stalks weren't very strong and the corn plant would just collapse very easily. Now suppose in Taiwan, there was corn being produced that didn't have very sweet kernels but had very, very strong stalks. Now you could essentially cross these two species and breed for the type of corn that you wanted that had both the sweet kernels and the strong stalks. This is the whole idea behind hybrid production of crops. It's interesting to note that hybrid corn was seen as evil in the 1920s. People said, well, this is not natural. You don't ordinarily, in nature, you don't mate corn from Iowa and Taiwan. Yes, pollen can spread very far, but it doesn't spread that far. And in fact, it took about 30 years for hybrid technology to be fully supported or to be considered socially acceptable in the United States. Today, all of the corn that we plant in the US is hybrid corn. That is the case in many industrial countries, less so in less developed countries. Now, fast forward to the 1980s, the whole concept of BT corn was invented. After basically the discovery of DNA in the 1960s and recombinant DNA in the 1970s, the idea that you could take genes from different species and then combine them into the same species to have a certain set of traits that you would want that organism to have. And in 1996, BT corn was sold for the first time in the United States. So I'll describe this more in detail later, but basically what BT corn is all about is that a gene is taken from a soil bacterium and inserted into the corn genome, such that the corn is able to produce its own pesticide. That is the whole idea behind BT corn. But now, what exactly is a genetically modified plant and how are they made? A GM plant is any plant that contains genes that we humans have inserted, rather than the plant acquiring the gene through pollination, which is the normal means of sexually, in a sense, trading different traits. And the genes can come from any species, not just the host plant. So for example, recently in agriculture, we've taken genes from a daffodil plant and inserted them into rice to produce what's called golden rice. This variety of genetically modified rice produces beta-carotene, which is a precursor to vitamin A. The idea behind the production of golden rice is that rice is a staple food in many parts of Asia, in many parts of Africa, and many parts of the world where people do not consume many other foods that contain vitamin A. So if there's a way to increase vitamin A production in a crop that's already being eaten by a lot of people, then that could help to prevent a lot of the diseases that are associated with a lack of vitamin A in the diet. And this is just one example of a GM crop. How are these genetically modified plants made? Well, first you have to decide, you as the individual, as a scientist, what is it that you want a particular plant to do or to be? Do you want it to produce its own pesticide? Do you want it to produce more vitamin A, vitamin C, vitamin E? Do you want it to be drought resistant? Do you want it to be resistant to a particular virus? Decide what trait you want that plant to have. And then you isolate a gene that you want from another species. It could be the same species, it could be a completely different species. Design that gene so that it can be successfully incorporated into what's called the host plant. So there are certain types of ways in which that particular gene is engineered by adding promoter and terminator sequences for the correct expression of the gene within the plant, and also a marker gene to indicate which are the plants that have successfully incorporated this transgene. Then you insert the gene into the plant cell through different mechanisms, and finally, from a plant cell that has successfully incorporated that gene of interest that you want, you can regenerate an entire plant by using plant growth hormones. This graph shows the acreage around the world that's dedicated to planting genetically modified or biotech crops worldwide from 1996 to 2007. Now, the green line shows the total planting of biotech crops over time, whereas the blue line shows planting in industrial nations, and the red line shows biotech crop planting in developing nations. So you can see that in the beginning, in the 1990s, most of the biotech crop planting was taking place in industrial countries. But now, as you can see, many developing countries are also planting biotech crops, to the point that now it's getting much closer to equal, that the acreage devoted to GM crops is close in both industrial and in developing countries. Now, it's not necessary to see all of the small lettering in this map. What I want to show you in this particular graph is where genetically modified crops are being planted around the world. Basically, they're in all the colored nations that you see that are not that sort of yellowish, beigeish color. So you can see it's planted all throughout North America and many parts of South America, many parts of Asia, Australia, and even in Europe. However, biotech crops are not being planted in most of Africa, as well as not in Russia and also in the Middle East. So now turning to the corn, or the variety of interest. What is BT corn? BT corn is the most commonly planted genetically modified corn in the US and around the world today. The term BT stands for Bacillus thuringiensis, hence the term BT. A Bacillus thuringiensis is a common soil bacterium. It's pretty much common in soil worldwide. If you just scoop up a handful of soil, there's almost certainly B. thuringiensis in it. This soil bacterium produces what are called crystal proteins. These proteins kill insect pests of the order Lepidoptera. However, they are harmless to other species. Scientists in the 1980s found a way to isolate the gene from the soil bacterium that produces the proteins, and to insert it not into just corn, but also cotton and potato, and today even rice, such that these crops are then able to produce these crystal proteins that are toxic to the insect pests that would chew on them. And the pesticide is contained within the plant. It's not just sort of scattered into the environment. Now, how is it that we can insert a bacterial gene into corn that kills insects but doesn't harm us and doesn't harm other species? Well, as I mentioned earlier, the genes produce what are called crystal proteins. You may well know that our digestive systems are very acidic. Among other things, in our stomachs, we produce hydrochloric acid. That's one of the strongest acids. I think it has a pH of close to 1. Many animals have very acidic digestive systems that basically break down these crystal proteins immediately. However, lepidopteran insects have basic guts. That is, the pH in the gut is higher than 7. And in those basic conditions, the crystal protein binds to the insect gut and causes a lesion in that gut. That causes the insect to stop feeding, and then the insect dies within a matter of hours or days and does not chew on the corn anymore and cause pest damage. There's a picture of Bacillus thuringiensis right there. Now, how does BT corn differ from the pesticides that would otherwise be sprayed on the corn? Well, the crystal proteins are very specific to particular species. So in fact, one crystal protein that might work to kill one insect doesn't actually kill other insects. So in fact, there are crystal proteins that are nearly 100% effective against all of these different corn borers and worms that you see listed here. But since they are so specific, now what scientists have done is to stack the varieties, that many different genes are inserted into BT corn to control for essentially all of these common corn pests in the United States. This is typically more effective than conventional pesticides, all because of the issue of how and when pesticides are sprayed. Pesticides have to be applied at exactly the right time in the field, otherwise you don't kill the insects. Basically, if you spray pesticides at the wrong time, they might not get the insects at the right time. They are easily blown off by the wind or washed off by the rain. And also, BT corn, the crystal proteins in BT corn are more environmentally friendly than other pesticides, and I'll describe that more later. What's this timeline for BT corn? It's actually quite interesting. The main pest in the corn belt, that is most of the Midwestern states of the United States, is the European corn borer. For about 80 or so years, the European corn borer has really destroyed a lot of our corn crop in the United States. Now, how did it get this name and how did it come to the US? Well, in the 1920s, we imported a lot of corn husk dolls. I think you may have all seen what corn husk dolls look like. You just basically take dried husks and you form them into dolls. We imported them from Europe, and the corn borer came along with it. And it quickly spread throughout the United States, the corn planting region, because it just happens to thrive in our environment, destroying a lot of our corn. In the 1930s, German scientists discovered that many dead worms in the soil, when they opened up their guts, they noticed the presence of this particular bacterium, Bacillus thuringiensis, and they thought, there must be something about this bacterium that's killing off these worms. What is it? As I mentioned earlier, in the 1920s to 1950s, hybrid corn became popularized in the United States. And then in 1961, because of the knowledge that was acquired in the 1930s, microbial Bt sprays were formulated basically to take the bacterium and to spray it on crops. Now, this is one form of an acceptable pesticide for organic growers because it's a natural source and it's relatively more environmentally friendly. In the 1970s, recombinant DNA technologies were discovered, that is, crossing genes from different species. In 1986, biotechnology regulation was essentially formulated in the United States. And as I mentioned earlier, in 1996, Bt corn was first planted in the US. Today, Bt corn planting makes up about 60% of the total acreage of our field corn. So basically, we pretty much are all eating Bt corn in all of our various corn products, corn chips, corn dogs, taco shells, you name it. Now, that's happening in the United States. What's happening around the world? You can see in this pie graph which nations are planting Bt corn. We're planting the vast majority of the Bt corn around the world, about 85%. But in fact, there are 9 nations worldwide that plant Bt corn listed there for about 25 million acres worldwide. And there are also field trials for Bt corn going on in China. Now, why is it that farmers worldwide want to plant this particular crop? Because they love the yield improvement. Before Bt corn was introduced in the United States and in other parts of the world, farmers had one of 2 options, neither of which was really desirable. One is to let insect pests destroy anywhere from 5% to 50% of their corn because of all the insect damage, as you can see in this photograph here. Or they could attempt to spray pesticides that occasionally worked and occasionally didn't, with some adverse health effects to the farm workers and also some adverse environmental effects. Now, Bt corn, because it protects against multiple insect pests, it's saving US farmers today, on average, anywhere from $20 to $200 for every corn acre. It does cost more than conventional seed. It costs about $8 more per acre, but as you can see, the benefits in this case outweigh the costs. So that's why farmers love it. But what's the impact to all the rest of us, our health and the environment? Well, first let's talk about who is paying attention to this issue and whether it's being regulated. So after these rDNA technologies came out in the 1970s, there was a lot of concern from a regulatory standpoint. What are the dangers associated with combining genes from different species? And particularly, what happens if we plant crops that have genes from different species? In 1986, the Coordinated Framework for Regulation of Biotechnology within the Office of Science and Technology Policy reviewed this issue, and they were interested in trying to figure out, well, do we in the United States need to create an entirely different regulation for these biotech products or for genetically modified organisms? Or do the existing statutes protect us adequately in terms of the risks? They decided that the existing statutes were adequate to regulate GMOs. Basically, that a new framework of regulation was not necessary for biotech. So today, as far as Bt corn, there are 3 agencies that share regulatory oversight and risk management. Those are the US Department of Agriculture, USDA, the EPA, the Environmental Protection Agency, and the FDA, the Food and Drug Administration. Now, how do they divide up this risk assessment? Well, the FDA in the US regulates both food and drug safety. In the case of genetically modified crops, the FDA made a decision in 1992 that genetically modified food is what's called substantially equivalent to non-GM food. And as a result of that, there is no mandatory testing of food safety. Rather, industries that develop biotech crops undergo a voluntary consultation process with the FDA. And also because of this ruling of substantial equivalence and the fact that it's also called GRAS, generally recognized as safe, there's no requirement for labeling GM food differently from non-GM food in the US. And that's why when you go to the grocery store, you pretty much never will see a product that's labeled contains GM ingredients, because there is no requirement for labeling by the FDA. The US Department of Agriculture regulates agricultural risks, on the other hand. So what USDA risk analysts are interested in looking at is, what is the potential for GM crops to cause harm to natural habitats or to agriculture in general? The EPA regulates pesticides, basically the toxicity, allergenicity, and carcinogenicity of different pesticides to humans and to animals, and they also conduct ecological risk assessments. So over the last 20 years, BT corn's food safety and environmental risks have been assessed in terms of what are the human health risks, what are the consequences of gene flow through pollination, what's the potential harm to other species, and what if insects develop resistance to BT toxins. So now let's go through each of these one by one. What are the human health risks? When we take a gene from another species and insert it into corn, essentially what that gene is doing is producing a novel protein that the corn would not otherwise have produced. What are the impacts of that protein? We have to be concerned with 3 categories of effects. Are there any toxic effects from that protein? Is that protein carcinogenic? Could that protein be allergenic? And a variety of different tests are carried out to determine whether any of these 3 categories could occur in terms of human health. Now, what types of tests? Mammalian tests are done, tests, for example, on lab rodents or on lab monkeys, to see whether the presence of the protein results in any toxic, carcinogenic, or allergenic effects. And allergenicity really is the main concern with proteins, because proteins are very rarely toxic or carcinogenic, but proteins are potentially allergens. And so there are 2 other types of tests that are done. The amino acid sequence of the introduced protein is assessed to see, well, does this amino acid sequence look in any way like the amino acid sequence of other known allergens? For example, allergens in peanuts or shrimp, or a variety of other potentially allergenic sources. Not only is the amino acid homology studied, but also stomach acid digestibility. So gastric conditions are simulated outside of the body, and the protein is basically inserted into the acid, and scientists observe, well, how long does it take for the acid to degrade that protein? If it takes too long, then there's some sort of concern that our bodies might take up that protein and that it might, in fact, produce an allergic reaction. Tests that have been done as far as the BT proteins have shown that there is no risk of toxicity or carcinogenicity, and that crystal proteins are non-allergenic. However, you may have heard of a case involving Starlink corn. Is that a term familiar to many of you? Okay, some nods and some head shakes. I'll explain to you a little bit about this. Starlink was formerly a variety of BT corn that was planted in the United States. In fact, in 1998, the Environmental Protection Agency approved Starlink corn to be used in animal feed, but not in human food. Now, why would that be the case? They were concerned that Starlink corn might cause allergens, because when they did the stomach acid test, they saw that for whatever reason, this particular crystal protein, Cry9C, was taking relatively longer to digest under the acid conditions. And so the company that was interested in selling this particular type of corn, Aventis Crop Science, was really pressing for EPA to approve it. And they made the approval that, well, right now we will allow it for animal feed. We don't really care about whether there are allergic reactions in animals, but we will not, for the time being, allow it in human food. And so EPA required that Adventist Crop Science, when they sold their seeds to farmers, they had to get a guarantee from their farmers, first of all, do not sell the corn that you produce for human food purposes, and also required certain planting distances. Well, in spite of that, 2 years later in 2000, when taco shells and corn dogs were pulled from the grocery market, traces of Starlink BT corn were found in them. How did this Starlink corn wind up in our human food when it wasn't supposed to be in our human food? Well, there's a long chain of potential human errors that maybe a farmer might sign an agreement, but he might accidentally sell his corn to a grain elevator that is sending its corn for human food uses. There might have been a mistake at levels of food processors that instead of the food going to animal feed, it somehow wound up in a human food bin. Or it might not even have been a human mistake. Corn pollinates. So sure, there can be a planting distance of 660 feet, but in the event of a very strong windstorm or rainstorm, the pollen can blow a lot farther than 660 feet. The pollen can wind up in a neighboring plot of corn. A farmer could be growing Starlink corn without even knowing that he is. And that's what some of the danger is. Our agricultural system is not foolproof. And these are the types of mistakes that can occur. And so we always need to be aware of them as far as any types of agricultural risks. Now, 1 year later, EPA decided that Starlink was not allergenic after all, but it was too late. Over $1 billion had been lost in terms of lawsuits, in terms of many other types of concerns, and Starlink corn was essentially pulled from the market, and Adventist Crop Science went out of business. And that really turned, so what I was talking about the pollen spreading a long distance, leads us to the risks associated with gene flow. What happens if non-BT corn or native races that are related to corn acquire these BT genes? Is there any sort of risk to the ecosystem? And we also need to be concerned about that. Particularly in areas of Mexico and Central America that are essentially basically the homes of many different native land races where we had originally bred our corn. If these different varieties acquire BT genes, does that give them somehow an unfair advantage that allows them to outcompete other species surrounding them? Well, this is a topic of concern in Mexico, which is why Mexico does not allow for BT corn planting. Nonetheless, 8 years ago, scientists at UC Berkeley, David Quist and Ignacio Chapela, found traces of BT in the Mexican land races. They had published their study in the journal Nature, which is a very prestigious scientific journal, but then that study had to be withdrawn because many people were protesting that the studies had not been done correctly. And indeed, there were some issues with the studies. They could have done it more rigorously, but probably their findings were correct. And in 2008, Alvarez Builla and her colleagues did do more rigorous testing, and they found BT genes in these native Mexican corn plants. And that indicated that farmers probably were planting it, even though it wasn't allowed. Now, what are some of the risks associated with this? Well, if corn acquires a BT gene, what that essentially does is that produces a particular pesticide. However, the problem is that, the issue is that many of these lepidopteran insects that are targeted by the pesticides are not really pests in these parts of the world. And so the risk is actually small, but there are very important cultural issues that if this is basically the home of native species of corn, maybe the people just don't want it to be contaminated with genetically modified material. And this is not something that we can lightly dismiss or ignore. Now, is there any potential harm to non-target species? Well, the Environmental Protection Agency conducted a thorough ecological risk assessment and showed that there were no harm to other species of insects, including bees, butterflies, moths, and ladybugs, no harm to annelids, such as earthworms, no harm to fish, and no harm to birds. However, there were originally concerns about whether BT corn would be toxic to the monarch butterfly. What happened in this case is this. Monarch butterflies do not normally consume corn. So how is it that they can ever come into contact with Bt toxins? Well, monarch butterfly larvae, that is when they're in the worm stage, they consume milkweed. And milkweed can grow fairly frequently around cornfields. And getting back to the issue of corn and pollen, the corn lands everywhere around the cornfield and can land on the milkweed leaves. Now, if the monarch larvae eat this Bt corn pollen, what's the impact to their health? A study led by Losey and his colleagues in Cornell, published in Nature 10 years ago, showed that 44% of monarch butterfly larvae, when they were force-fed Bt pollen for 4 days, 44% of them died. Now, this caused a lot of concern because if there's any risk of planting Bt corn to monarch butterflies, this is obviously not something that we want in society, because as a society, we value having monarch butterflies. So a series of follow-up studies were done over the next 2 years to figure out, well, in real life, do monarch larvae really eat this much Bt corn pollen? And the results of the studies, which were published in the Proceedings of the National Academy of Sciences, showed that there wasn't really a significant risk, neither in terms of the effect nor in terms of exposure, because monarch butterfly larvae don't encounter, in fact, that much Bt corn pollen to cause any adverse effect in real-life conditions. Now, what if insects develop resistance to Bt toxins? You may all be aware of the fact that many insects over time develop resistance to pesticides. Now, how can that be the case? Well, if you apply a pesticide across a cornfield, many insects will die, but there will be some that still survive because they're somehow better able to resist the toxin. Well, the survivors will reproduce with each other, and they will produce offspring who are also resistant to the pesticide. And over time, these resistant species will basically take over the cornfield. And if you apply the pesticide, it's not going to be useful anymore. It's not going to kill these insects. Now, this is also potentially a concern with regard to Bt corn, that over time, insects may become resistant to these Bt proteins. Now, this is particularly risky because we have to think outside the box. We have to think not just about whether insects become resistant to Bt corn, but remember that I said earlier that organic farmers use a microbial Bt spray. If insects develop resistance to Bt corn or Bt cotton or Bt rice, then they may also have resistance to these microbial Bt sprays, which means that organic growers may not be able to use this microbial formulation anymore. And that is a serious concern. In fact, no insects have developed resistance to Bt corn yet, but this is something that we always have to be thinking may potentially happen. The EPA has what's called an insect resistance management, or IRM plan, that basically involves what's called a high dose and refuge strategy. That is, the dose of the crystal proteins is extremely high to try to kill as many pests as possible. And also refuges, non-Bt corn, are planted such that there can always be susceptible pests to mate with any potential resistant pests that might produce susceptible offspring. And the idea is to delay resistance as long as possible. And there's also insect resistance monitoring going on constantly to see if there are any resistant insects, and if they are, then Bt corn planting will be stopped. So much for all the risks. Now let's talk about some of the potential health and environmental benefits. I'm going to talk about 2 categories: reduced use of pesticides and reduced mycotoxins in our crops. Basically, less of this spraying going on and less of the damage that's caused by the mold that you can see in these pictures. So Bt corn reduces the use of sprayed pesticides. Why does this matter so much? Because the conventional pesticides, the chemical organophosphate, carbamate, pyrethroid pesticides, are toxic to far more species, non-target species, than the insecticides, the crystal proteins, that are found in Bt corn. Not only that, but 98% of the sprayed pesticides don't even reach their intended targets. They land everywhere else. Humans are exposed to them, especially farm workers. They land in the soil, they get into our water system, and they affect wildlife everywhere. I mean, this is not too hard to imagine. I mean, you can see this plane here spraying the pesticide all over the crop. I mean, some of it will reach the crop, and then some of it will reach the insects, and then the rest will be scattered all over the soil. When Bt corn is not planted, corn growers typically use these 4 pesticides: methylparathion, chlorpyrifos, permethrin, and lambda-cyhalothrin. In humans, 3 of the 4 of them are extremely neurotoxic, and some of them are endocrine disruptors, and some of them may cause other adverse effects. Among wildlife, all of these pesticides are highly toxic to fish and other types of marine wildlife, and some of them are toxic to birds and others are toxic to rodents. Now, what has happened now that Bt corn has been planted? What's happened to the planting of these pesticides? What you see in these 2 graphs to the right is Bt corn acreage by year from 1991 to 2002, and the bottom graph shows the planting of those, or the use of those 4 pesticides that have the neurotoxic effects to humans and also the toxic effects to animals. You can see that, well, Bt corn was not planted until 1996. And before that, there was a certain rise and fall with different pesticides used. But as soon as Bt corn planting went up, the use of those other pesticides went down. And then when when Bt corn was used a little bit less in 2000 and 2001 because of the Starlink debacle, the use of the pesticides increased again, and then they went down again. Now, there are some caveats to this. 2002 is the last year for which I was able to find data on this. And the reduction in pesticide use could be caused by factors other than Bt corn, but it's reasonable to suggest that there might be an association between these 2 factors, that by planting more Bt corn, you're reducing your pesticide use and thereby reducing the risk of harm to the environment and to human health. Bt corn also reduces fungal toxins. Mycotoxins are the main area of my research right now. The fungi that colonize our plant crops can produce toxins. If you own a garden, you may have seen this yourself, that insect pest damage is one of the major ways in which fungi colonize our plants. For example, if you grow tomatoes, you might see that where there's insect damage, there might be a little ring of mold around the insect damage or even inside it. And the reason is that insect damage basically opens up the crop and exposes the sugars. The fungi love these sugars. They quickly colonize the plant, and in many cases, they also produce toxins. Now, these mycotoxins can cause acute illness as well as cancer in human and in animals. Bt corn, because it has significantly less insect damage, has been shown in field study after field study to have significantly lower mycotoxin levels. Now, this is a little-known secondary benefit of planting genetically modified corn with different types of benefits. In the United States, farmers benefit because they can sell their crops without all this mold and not have to worry about it being rejected. In less developed countries, and this is where it becomes really important, the consumers benefit the most by reduced fungal toxin damage. How bad are mycotoxins? Here's a historical review. I wonder if any of you have even heard of mycotoxins before. Is this a term that any of you have heard of? Some of you. Okay. So fungi produce mycotoxins under particular environmental conditions. These chemicals, these mycotoxins, have toxic and carcinogenic impacts in humans and in animals. The history of mycotoxins in our food dates back to the 11th century in Europe. In the 11th century in Europe, people were not yet planting wheat or planting corn. Rye was the main grain that they were consuming. They used this in their bread, they used this in porridge, and in other types of cereals. Rye is oftentimes colonized by a fungus, Claviceps purpurea, and this produces toxic ergot alkaloids in the rye. At the time, there was a disease called St. Anthony's fire that was widespread throughout Europe. It was a really nasty, gangrenous disease in which basically your fingers and your toes shrivel, turn black, and you also experience hallucinations. This was caused by those ergot alkaloids in the rye that people were eating, but nobody really knew why at the time. Instead, the disease was called St. Anthony's fire because people believed that making a pilgrimage to the shrine of St. Anthony in what's now present-day France would cure them of their symptoms. And indeed, that is what happened. When they made that pilgrimage to that part of France, their symptoms miraculously disappeared. Now, that may not be so much as a result of praying, but What happened actually was that the church in that particular area of France ordered farmers to sieve out the black grains of rye and to only make the bread and the flour out of the grains of rye that were not black. They didn't know it at the time, but they were basically getting rid of the mycotoxin in the rye, and so the quality of the food in that part of France was a lot cleaner, and the disease symptoms disappeared. But people didn't know why. Fast forward to another 1,000 years, in the 1930s. During the Great Depression era in the United States, there was a massive drought across much of the Midwest. Many farmers who planted wheat ordinarily turned to planting corn, because corn is relatively more drought resistant. Unfortunately, under drought conditions, corn is very easily colonized by another fungus, Fusarium verticillioides. This produces a mycotoxin, fumonisin, that is implicated with esophageal cancer in humans and also neural tube defects in human babies. It has a variety of other adverse effects in animals. During the 1930s, veterinarians found that many horses who were consuming now corn instead of wheat were dying of a mysterious brain disease, and they didn't really understand why at the time. At the same time, in the 1930s, many human babies across the US that were born in that decade had neural tube defects, and nobody really understood why at the time. In fact, mycotoxins were not discovered until 1960. The first mycotoxin discovered was aflatoxin, in connection with a large number of turkeys that died in the United Kingdom from consuming peanut meal that had very high levels of this toxin, produced by the fungus Aspergillus flavus. Today, we now know about many different mycotoxins, and I'll just talk about the ones that might be of concern to you in the corn. Fumonisin, aflatoxin, vomitoxin, and zearalenone. I'm not going to quiz you on the names of these complex mycotoxins, but what I really do want to emphasize are what their health effects are. As I mentioned earlier, fumonisin causes neural tube defects in human babies and is implicated, although not proven, to cause esophageal cancer as well. It causes a variety of adverse health effects, including brain diseases in horses, pulmonary edema, a lung disease, in swine, and also a liver and kidney cancer in rodents. Aflatoxin is the most potent natural human liver carcinogen that's known. Basically, it's, if you consume large amounts of aflatoxin, you could basically shut down your liver and die as a result. At chronic low doses, it's a very strong risk factor for liver cancer, and it also suppresses our immune system. I don't think I need to tell you what vomitoxin's impacts are. You can tell by the name, and this has in fact, well, DON obviously causes gastrointestinal upset in a variety of different animal species. Zearalenone is what's known as a mycoestrogen, and that you can see from the molecular structure, it very closely mimics the female hormone estrogen. And so it can essentially displace estrogen in the body and cause all kinds of odd endocrine disrupting effects. Now, why does BT corn reduce mycotoxins? I began to allude to this earlier. Because BT corn has less insect damage. And as a result of that lower insect damage, there's less fungal colonization of the corn, and subsequently, less mycotoxins in the corn. Now, what is the field evidence for this? A variety of studies that were done coming out of Iowa State University showed that the varieties of BT corn that are still being planted today, BT11 and MON 810, have significantly lower levels of fumonisin than non-BT corn varieties. As I mentioned earlier, fumonisin is the mycotoxin that causes the neural tube defects. BT corn has significantly lower levels. And this result wasn't just found in the United States, it was also found in many different countries around the world. France, Italy, Turkey, Argentina, Germany, and Philippines. So far, the evidence is that wherever BT corn is planted, fumonisin levels are significantly lower. There's a new variety of BT corn that will soon be sold in markets that has significantly lower levels of aflatoxin. Now, that can be shown to have very significant health impacts around the world. Varieties of BT corn that are already being planted also have lower levels of vomitoxin and zearalenone, and this is especially beneficial in animal feed. So that's certainly beneficial in the United States, but more in terms of benefits to farmers, because we have a very good regulatory system to protect us from mycotoxins. Basically, the Food and Drug Administration monitors how much is in our food and basically rejects food that has very high levels. That is not happening in many less developed countries. There's a lot of what's called subsistence farming in less developed countries, that people are simply consuming what they grow. And many times the corn and the peanuts that they grow never enter any sort of regulatory inspection for these mycotoxins. And many times also in less developed countries, if there's a food shortage, even if the corn or the peanuts are obviously moldy, people have no choice but to eat it anyway because they have to eat. And so in many parts of the world, there are very, very high levels of aflatoxin in the food. And it may be that if BT corn is planted there, by reducing aflatoxins, you could significantly reduce the risk of liver cancer, which is endemic in less developed countries. How much so? Liver cancer, you may not hear many cases about liver cancer in the United States because in fact there aren't that many, but worldwide liver cancer is the third leading cause of cancer deaths, following lung cancer and stomach cancer. The rate of liver cancer is 16 to 32 times higher in less developed countries, and aflatoxin is a major cause of this because there's a lot more aflatoxin in the corn and peanuts. Not only are the corn and peanuts more contaminated, but people are eating a lot more corn and peanuts, particularly in sub-Saharan Africa and in China. If BT corn can reduce aflatoxin, that can reduce some of the risks associated with this type of corn, with liver cancer. Well, if this is such a benefit, why don't we hear more about it? I wonder how many of you have even heard about this potential impact before. Well, there are several reasons for that. As somebody had asked earlier, well, why aren't many countries yet planting genetically modified crops? One reason is that the benefits of genetically modified organisms are not part of the regulatory decision in many countries. They just assess the risks. Benefits assessment is not something that's a part of a lot of this regulation. There are also risk communication issues that in industrial countries, including in the United States, we don't hear much about mycotoxins. Generally, this is not an issue that food processors want you to hear a lot about because there's no need for what might be considered an unnecessary scare. And also, in many poor nations, BT corn and other GM crops are not even yet accepted. So even though we want to communicate what the health benefits could be, there are still multiple barriers in the way. Now, the final portion of my talk concerns BT corn in the context of current events. I mentioned before that corn is really an important factor in agriculture today. It's going into our human food, it's going to feed our animals, now it's going to feed our cars as well, and this is becoming more of an issue worldwide. What are some of the issues? Meat consumption is increasing in China and India. Now you might ask, well, what does that have to do with corn? Some of you may know that in order to produce 1 pound of meat, whether it's chicken or pork or beef, requires 10 pounds of vegetable to feed to the animal that you're feeding to create the meat. And around the world, this averages out to 9 pounds of corn and 1 pound of soybean meal for every additional pound of meat that's being consumed or produced. The middle class is growing in China and India, and these middle classes are demanding a more Western lifestyle, which includes consuming more meat. What are some of the statistics of what the implications of this are? In China, there's 1.33 billion people. In India, there's 1.15 billion people. These two nations together make up more than 1/3 of our total world population. Now, if we were to take a conservative estimate and say that each person in these nations ate just 3 pounds more meat per year on average, and my husband can eat this amount in 1 day, then you would need an additional 67 billion pounds more corn annually, and that's 1.2 billion bushels, or 10% of US corn production. But we can't supply them with that corn. We're taking all our corn and we're making it into ethanol. In the United States, 98% of the ethanol that we produce comes from corn. In 2007, we produced 6.2 billion gallons of ethanol. We needed to use 13% of all of the corn that we produced in order to produce that amount of ethanol. But now the National Corn Growers Association has set a US goal called 15 times 15 times 15, that this isn't enough ethanol. We need to be producing 15 billion gallons of ethanol from about 15 billion bushels of corn by the year 2015. And that's expected to take up 24%, or nearly a quarter of all the corn that we're producing in the United States. Now, that might lead to some food and environmental concerns. From the food standpoint, do we put the global food supply at risk by using our corn to make fuel rather than making food? And from the environmental standpoint, by producing more and more corn, how do we affect our environment? Some of you may know that many farmers traditionally rotate corn with soybeans in alternate years. Part of the reason is that corn takes up nitrogen from the soil, whereas legumes, like soybeans, replenish nitrogen into the soil. But now, because of this heavy incentive to produce ethanol, there's a fairly high price paid to farmers for corn. So instead of rotating the crops, they're planting corn after corn after corn after corn, year after year after year, instead of the traditional rotation. Now, what's the impact to our environment from that? And to give you some perspective, 1 bushel of corn, which is about 56 pounds of corn, if you can imagine the weight of maybe an 8-year-old child, yields just 2.7 gallons of ethanol, so a little less than 3 jugs of milk worth of ethanol. How can BT corn help to mitigate some of these risks that we're dealing with currently? Less insect damage means effectively a much better yield. And this is important both from the standpoint of ensuring our food supply as well as for our energy demands. The Bush administration was very pro-ethanol, and interestingly, the Obama administration is too. There's a real push to just use ethanol for fuel purposes and to produce more and more corn to basically be a feedstock for that ethanol. Both administrations support this practice, and we need to meet it. BT corn also leads to lower pesticide use, and that leads to improved soil and water quality. This is less of a concern than if we have to keep on planting more and more corn because of the impacts on the ecosystem. If BT, as a result of all this massive corn planting around the world, catches on in other nations, then aside from the benefits as far as yield, there could be lower mycotoxin levels in the food supply, which could essentially benefit everybody. To summarize all of the points that you heard tonight so far, well, BT corn is just the latest development in 11,000 years of humans manipulating and domesticating corn. So far, we do not have any evidence of food or environmental risks, but there is a fair amount of field evidence of food and environmental benefits in terms of pesticide reduction and mycotoxin reduction. And because of the current agricultural demands for ever more corn production today, BT corn may play a very important role in current events, including in energy policy and in the food supply. At this point, that is the end of my presentation, and I'm happy to take any questions. Thank you all.
So your comments about improving food safety because the mycotoxins are quite compelling. But the question that I have is a smaller one, and that is, given how very little we know about food and food allergies, is the statement that there's no evidence of a food allergy actually a pretty weak one? Do you understand my question?
Okay. The question that this lady asked is that with regard to food allergens, the fact that there is no evidence of food allergenicity, does that in fact mean, well, does absence of evidence mean evidence of absence, I think is the essence of what you're asking.
Especially with what I believe is a true statement, which is that we know very little about food, how food and food allergies work. As a scientific community, we know very little.
Okay, well, I can explain a little bit about allergens, and excuse me if I get a little bit technical here. The reason that some people are allergic to particular proteins and other people are not, it all has to do with how our immune system responds to the protein. Basically, when our immune system recognizes something that it perceives as being toxic, it can result in some sort of overreaction, an overreaction to an immune response. So you may be familiar with some allergic reactions associated with food items, like hives, like anaphylactic shock. some of these other impacts. It all essentially is an immune system reaction. We know a fair amount about what in proteins can cause allergens because we have a very, very large database of all the food allergens, and there are particular amino acid sequences that are associated with allergenicity. What's been done as far as looking at BT corn and other GMOs and the risk of allergens are three different things. That there are mammalian tests to see whether there's any evidence of allergenicity in animals. But as you know, that can only go so far because animals are just like humans. There's a lot of variability. I'm allergic to shrimp. Many of you probably are not. So if all of you got tested but I didn't, then that doesn't give you much information. So in addition to those mammalian tests, other tests are done. For example, the amino acid sequence of the protein is examined to see, well, are there any amino acid sequences that look very similar to the amino acid sequence of all the other known allergens? And if there is, then that is a source of potential concern. If there is not, then another type of, a third type of test is done in any case, which is basically to simulate gastric juices, to submerge the protein in these juices and see how long it takes for the acid to degrade the protein. Because if the protein is degraded just fine, then it is not an allergen. Basically, it just becomes a separate amino acids and the human immune system does not respond to it. These are the types of tests that have been done for the crystal proteins. And if just speaking from my own personal standpoint, if only mammalian tests were done, I would be skeptical. If the amino acid sequence were done, I would feel a little bit better about that, but I really feel the best about the gastric acid tests to see whether those proteins can be quickly degraded, because then there's not really much risk of the protein causing an immune system reaction. Yes, sir.
Just following up on that, because I've seen some scientific evidence that it's going the other direction. And are you familiar with the Terje Traavik?
I've heard his name, yes.
And he's the director of the Norwegian Institute of Gene Ecology. And just two things to bring up and give you a comment on both of them, I appreciate. He's a very well-regarded scientist. He had hundreds of papers, published on his own. But he's done studies on this, a review study, and he cited 14 different scientific studies that were connecting BT with allergic reactions or possible toxic reactions, some done on mammalians, some on humans. He did some himself in the Philippines in 2003. About 100 people living next to a BT cornfield developed all kinds of CN, respiratory, and intestinal reactions. And this repeated itself in 4 different villages, other villages in the Philippines. Vomiting, headaches, things like that. He did blood tests himself on 39 people. 39 people showed an antibody response to BT toxin, which doesn't prove, but it does support a link. So that is one. And he's also, so he did work on his own, he was also citing 14 other scientists. The other, one he did not cite, because it just happened, it just came out a couple of months ago, was that a BT corn, this is the study done by the Austrian government. And BT corn, they gave rats one kind of GM corn that contained BT, and other that didn't. And the rats that were getting it, the 3rd and 4th generations down, were showing that not only were they having fewer rats in the litter, but the ones they were having were lesser weight. And this is widely reported in Europe and not here. And
Can you ask for a question?
that was, yeah, if you just comment on those two, that is my question. Can you comment on those two, both Traavik and his findings, and then this Austrian study that came out a couple months ago?
Okay, sure. I did hear about both of those studies. As I understand it, that particular study that was carried out in the Philippines, I believe that he published it in public sources, but not necessarily in a peer-reviewed scientific journal, that particular finding. I think in those particular cases, it wasn't really clear whether, whether it was exposure to BT or to some other agricultural factor. And in that type of study, you have to develop what's called a mechanism of action or a mode of toxicity. If it is the BT protein, what is the mechanism by which it's causing all of these particular adverse impacts? Now, as I understand it, he never was able to establish a mode of toxicity. And he did not also control for the other factors that may have caused the headaches, the nausea. And had that kind of study been done, then it could have been published in a peer-reviewed scientific journal. With regard to the Austrian study, that, I confess, I have not followed up on. I thought that was a Roundup-ready variety, not a BT variety. It was both. Okay. And there too, I would be curious as to what the mode of toxicity is, because now you're talking about several different proteins. If that is indeed the case, that there was a difference in basically the weights of the third and the fourth generation pups, then you would have to figure out, well, what is the mechanism of action that is causing this reduction? And then you would better be able to understand whether the protein was responsible for it. So it's an intriguing study, and that's one that I would have to look more into to basically give my own scientific judgment on. Yes?
I was curious about the availability of seed for BT corn and whether it was something that, like, some company, like, I don't know, Monsanto or somebody owned the rights to distribute the seed, or whether it was something that was distributed, how it works.
Okay, that's a good question, and I'll repeat it to the audience. This lady asked, well, basically, in essence, who owns the seed? Is it a company that owns the seed, or is it something that we all have access to? At the moment, yes. Companies such as Monsanto, Syngenta, Pioneer Hybrid, they have patents on the gene transformation, what they call a BT corn event. And what that means in practice is that there's what's called a technology fee that's applied to the seed. So when I mentioned earlier that BT corn seed costs more than conventional corn seed, that is at least part of it, that companies have intellectual property rights over these seeds. And aside from just the cost issue, there's also the issue, which I really do think is a potential problem in less developed countries, that when these seeds are planted, farmers cannot just save the kernels from previous seasons and then plant them the next year. They have to buy new seed every year. And that is not that's something that farmers in the United States have been doing for 8 decades. We're used to it. We're used to planting the new hybrid corn every year because we want all our corn to be uniform, about the same height, to all be flowering at the same time. But that is not the case in agriculture and many other parts of the world, where they're still saving the seed and planting it the next year. So there are a lot of issues to be overcome with that regard. How much would these companies charge poor farmers to plant the seed? Would they waive the rights as far as farmers saving the seed and being able to plant it next year? All those things still need to be worked out. Yes?
So if the USDA generally recognizes as safe the BT corn, the studies on the safety, are those conducted by the company submitting the GMC? Who does the testing? Are they basically saying, yes, we believe it's safe, and the USDA stamps it off?
Okay, the question that this gentleman asked is, okay, given that, well, it's actually FDA that made the ruling of grass, or generally recognized as safe. Who is conducting these safety studies? Is it industry, or is it somebody outside of industry? Now, there's actually a different study, there's different cases for different genetically modified crops versus BT corn. Because in the case of BT corn, EPA and other scientists have also done their own independent studies. But with regard to GM crops more generally, it is industry that conducts the studies, and then scientists from the FDA and EPA look at how these studies were conducted, look at the results, and make the assessment of whether there's enough evidence of food safety that it can be allowed for planting. Excuse me. Are there any other questions? Yes.
So I was wondering, so are there ecological safeguards against like the pests that feed on the corn? if you made the corn cancer resistant, it seems like you're sort of cutting something out of the ecosystem there. Have we thought about that, like 2 or 3 steps further down the line?
The question that you're asking is by the fact that the corn is producing the pesticide and the insects are consuming the pesticide and being killed as a result, does that affect the ecosystems?
Yeah, but like, yes, there's probably something else that eats the pests.
Oh, I see what you're saying.
Yeah.
Okay, yes, I understand what you're saying. That kind of study has been assessed in the past. For example, is there any risk, for example, if a European corn borer consumes a certain amount of Bt corn and before that insect dies, another insect might consume? And what happens to that particular insect? Well, tests have been done. For example, you might have heard about the tests regarding the green lacewing moth that may consume some of these lepidopteran insects. And so one of the issues with regards to these crystal proteins is that they're very specific to a particular species. So in fact, there's just like a few types of crystal proteins that are toxic to the European corn borer, but are not toxic to the other insects or other species that might consume the European corn borer. So if another species does consume the European corn borer that has the crystal proteins, that animal does not necessarily have a toxic reaction to the crystal proteins. Yes?
On that, if that natural pesticide kills the European corn borer, and you're removing the corn borer from the food chain, has that study been done? So it's sort of a different variance of the same question. And if I ingested it, or the corn borer ingests it, somebody eats it before it dies, then you're preserving that food chain. But if the pesticide kills the corn borer before it becomes a food source, it dies, you're removing that element from the ecosystem. What happens?
Okay, I think I understand your point, and I'll repeat it to the audience. Aside from the issue of whether there's any harm to whatever might consume the European corn borer, the fact that there might be less corn borers as a result of Bt corn planting, does that have some sort of effect in the entire food web of the ecosystem? That's what you're asking? And that's a really interesting question, because there might very well be an impact. That I don't know. On the other hand, the European corn borer was introduced to the US in the 1920s. Before that, we didn't have the European corn borer. So there may indeed be some level of disruption in the ecosystem. The question is that more so than if we were applying the methylparathion and permethrin and chlorpyrifos to the ecosystem? There's lots of uncertainties. Yes?
At what stage in organic farming is the Bt spray applied? It's to the ears. So when we eat organic corn, is there Bt residue in the organic corn?
So the question that this lady asked is, well, if the microbial Bt sprays are used in organic crops, is there a residue on the crops that we are consuming? Actually, those Bt proteins degrade very quickly. I mean, that's one aspect of the microbial Bt sprays, that it has to be applied at exactly the right time, because it actually does not linger in the environment nearly as long as some of these organophosphate pesticides. So likely by the time that we consumed the corn, we probably wouldn't be ingesting the microbial Bt spray. Yes?
How stable is the Bt that's in the plant?
This lady asked how stable is the I guess the crystal proteins, you mean? Okay. Well, so how stable are the crystal proteins that are that the Bt corn is producing? Pretty much the proteins are being produced all the while that the corn plant is still alive. But toward the end of the corn plant's life, whenever it's about to be harvested, usually in like early fall, then the crystal proteins, it produces less of the proteins. But the protein is present all the while that the corn is growing.